Everyone, and welcome to my YouTube channel. So here today we are going to be looking at the introduction to genetics. So to begin with, what is genetics? So genetics is simply the specific or scientific study of genes and heredity. So now heredity, which is heredity, is simply defined as how certain qualities or traits are passed from parents to offsprings, from one generation to the other. So this is what basically genetics is all about. So from one generation to the other. So that is what basically genetics is all about. So now, when we refer to genetics, we cannot talk about genetics without giving credit to a very important man known as Gregor Mendel, who is known as the father of genetics. So now Gregor Mendel is the father of genetics, and one examinable question that they might ask you is, what did he use? So he used what we call the Pisum sativum. So Pisum sativum is simply what we are going to call garden peas. So Pisum sativum is garden peas. So now the question that they might ask you is, why did he use garden peas? So number one, the first reason that he grew garden peas is, as you can see, they are very easy to cultivate. So that's reason number one, very easy to cultivate. And the other reason, they are very easy to cultivate and produce many distinctive varieties. So this is what we mean. So garden peas, some are round, some are wrinkled, we have yellow, it can come out green, meaning to say these are the many varieties. We have the inflated, we have the constricted, we have green, we have yellow, we have purple, we have white, this terminal, axial, we have tall, short. So it has got many varieties, so it makes it very possible for it to be a very good specimen for us to study the behavior of these peas. So these garden peas have got a very short generation as they grow fast. So now, why is this very important? So if you look at generation, it's more like from when it is very little, it grows, it's able to now produce the next generation, or rather the next two successors to also grow like that. Now they are very short generation. Like for example, the generation of human beings is about 30 years. That is the proper time that we expect the men to more like repopulate and all that. Now these have got very short generations, meaning it could be about 8 to 10 weeks. That's basically about just 2 months. So they are able to grow, which makes it very possible for us to study how each trait is affected in different conditions. So this is the cycle. This cycle is very fast, like that. Now it is important that this cycle should be very fast, so that we are able to see and determine what are the properties. So he couldn't have used, let's say, specimens such as humans to study the behavior, because already, or another animal, that would take 30 years for it to more like have the next generation. You would find that maybe you just study 2 times, you just study the organism's outcome 2 times, you even die. So he needed something which he can be able to do over and over to produce the outcome very fast. So that is why he used garden peas. So the other important thing is that sometimes he was required to study the behavior if the flower could cross, when it cross pollinates. So now it is very important This was made possible because both the male and female are enclosed, let me write it, within each P. So just as we have said, they are enclosed within each P. Flower and gametes are produced by the male and the female are able to like fuse, or rather merge. So as we can see, we have the male and the female part enclosed in one P like that. So the male producing the anther, so these can simply go on the stigma, then they can easily move like that. So they are enclosed like that. So as you can see, there is something that is enclosing them, making self-pollination to be very, very possible. So that's why he chose garden peas. Now let's define some terms that are used in genetics. So a gene is a section of chromosome that codes for a trait. Okay, let's begin with the word chromosome. Chromosome, it is a thread-like strand that stores genetic information. Now, a gene, a gene is a section or portion of a chromosome that codes for a trait. So the one that codes for a portion, it is a section or portion that codes for a specific trait. So a trait might be a characteristic such as height. So that is basically what a gene is. So I'm going to define the three terms, then I will bring in a diagram. Then when we look at the allele is an alternative form of a gene. So as we have said, an allele is an alternative form of a gene controlling a specific trait. Now, a specific trait of a gene. So now let me explain it with a diagram. So let's say we have alleles like that. So these are alleles. As you can see, they haven't written the word allele, meaning to say there are two. We have this one and this one. Now these together, our A and another alternative A, this together now makes what we call a gene. So a gene is a portion on a chromosome. So as you can see, this is a chromosome like that. Then the chromosome, then we have alleles like that. Then the alleles together will now form what we are going to call a gene. So a gene is more like the combination of two alleles. Chromosome, alleles, then those two alleles will form what we call a gene. Genotype, on the other hand, it is the genetic makeup of an organism. So that is what we call the genotype. So the genotype includes both the genes in homologous pairs. So homologous pairs simply means to say the portion of the chromosome carrying the same gene like that. I'll explain of chromosomes. So let's refer back to our image. So as we can see, these are homologous chromosomes. They are carrying the same characteristic here. So at that point, so the genotype can be written. So the genotype shows you the alleles that are present. So in this case, it is A and small letter A like that. So these are the alleles present in the genotype like that. While phenotype, on the other hand, we have the phenotype is the outward expression. So the outward expression of a gene. So that is basically what phenotype is. It is just the outward expression of a gene. Okay, to understand this, let's look at this analogy. We have this small book, we have this big book. So we can call this one, though we haven't discussed it, we can call this one as the dominant, then this one as the recessive. We are going to use the same idea. So now this one, it means to say, as you can see, now the phenotype is the outward expression of a gene. Now let's say if I was to put this book at the back. So as we can see, if there is a small book at the back there, then that simply means to say it won't show. So that means to say what you are able to see is what we call the phenotype. Now with this same idea, let me use it to explain what we mean as dominant and recessive. So dominant is the characteristic of a gene to mask another. So we have the characteristic of a gene to mask another, while recessive is the characteristic of a gene to be masked. So to be masked. So get the difference. So this one is to be masked, this one is to mask, or the characteristic of a gene to be expressed. Mask, or not masked but mask another. For this one it's to mask another. For this other one is to be masked, or rather not to be expressed if a dominant allele is present. So now as we can see, if the big book is dominant because it is able to mask the other book, as you can see, even if the book was at the back, you cannot be able to see it. That is what we refer to as the word dominant. So that is basically, just as we have said, dominant is the characteristic of a gene not to be of a gene to mask another or be expressed over the other. And also recessive is the characteristic of a gene to be masked, the one that is masked, the one that is not shown, or rather not expressed. That's the characteristic of being recessive. Other terms that are used in genetics, we are going to be looking at terms such as codominance. So we are going to look at codominance. So this one is the characteristic of gene to be equally expressed. So just as we have said, the characteristic of a gene to be equally expressed in the phenotype. So that is what we call codominance. The simple example that we might have is blood group AB. So blood group AB is an example of codominance, where we have blood group A and blood group B. When they merge, they are going to form our AB. Now these are equally expressed, so meaning to say that is what we refer to as codominance. They just from the word co means to exist, or coexist means to exist together. So codominance, they are equally expressed like that. Then when we look at the other example that we might have under the codominance is, so you should know the example. So we have blood group AB. They might ask you in an example. The other example that we might have is a checkered chicken. So we have the checkered chicken. So this chicken is formed as a combination of the white chicken and the black chicken to produce this color. As you can see, it has got white, black, white, black, white, black. That is what we call codominance, when the colors are equally expressed like that. So those are some of the two examples that you might have under that one. The alternative is what we are going to call incomplete dominance. So incomplete dominance occurs where the allele is not fully expressed, or is not fully masked like that. So the resulting offspring is an intermediate of the two. So this is what we refer to as incomplete dominance. This occurs when the allele is not fully expressed or masked, but the resulting offspring is an intermediate. So like for example, we could have what we call the roan cow. So we can have roan cow. So this is the roan cow. As you can see, we have the red or brown bull fuses with a white cow to produce an intermediate color which is pink. Neither of them is fully expressed, so that is what we refer to as incomplete dominance. Incomplete dominance is also seen in what we call the snapdragon, which are just flowers. So snapdragons. So as we can see, we have the red combining with the white to produce the intermediate color which is pink. So this is incomplete dominance. Let's look at our last example. The other breeds that we might talk about is the Andalusian chicken. So this one is just a combination of sometimes when the white chicken combines with a black chicken, instead of having the checkered chicken or something like white or just black, we might have the Andalusian chicken. This one shows an intermediate of black and white, but it does not favor any color. It's more like we have a slake. It's appearing a bit gray. It's appearing a bit gray like that. So this is what we call incomplete dominance. Just one last term, we have the alleles. We might have heterozygous, simply means to say the existence of two paired alleles, two different paired alleles like that. So as you can see, even the color pink is different from the color green there. So that means to say this is what we are going to call heterozygous. It is made up of two different alleles. Homozygous alleles means alleles of the same, carrying the same characteristic. If it's height, if it is tallness, then it is tallness. Then if it is just shortness, it's shortness, meaning to say small letter t, tallness, tallness. This is homozygous. Heterozygous means we have a big letter and a small letter like that. So that is basically what we look at under the introduction of genetics. Thank you very much for watching. Hope you would like, subscribe, and share for more. See you in the next tutorial.